All right, so to get back to the discussion of what a rock can do, the kind of chemistry that a rock can do. Well, we finished up with this slide right here, where we talked about iron-sulfur complexes. And all of these iron-sulfur complexes are actually seen in actual proteins. And the thing is, if you show this to geologists, they'll say that looks like the most common structure of iron and sulfur in rocks. And so they really see a lot of similarities there. The only difference to any of those is that you actually see molybdenum in one of them. You see the Fe MoCo that they have here. But this also looks like a rock. It's just sort of an arranged rock, okay? So you can imagine bits of rock like this floating around doesn't even really need a protein that much if it's doing the chemistry. And so it's possible that these were the first enzymes. Again, remember, not much oxygen around, and so a lot of sulfur around when there's not a lot of oxygen around, and you have even more of these floating around. So they're in fact, they're the major soluble form of iron in sulfur-rich seawater. So you look inside a hydrogenase, and this is from an ancient hydrogenase enzyme. And hydrogenase just means literally what the asteroid was doing. It means it's moving electrons onto or off of H2. In a biological system, that might be onto or off of an H2 substitute like NADH or FADH2. But it's really the same kind of thing. You're just moving electrons. And so this is an early hydrogenase enzyme, and you notice a lot of things. There's 2 irons, there's a bunch of sulfurs, there's some bridging atoms, and if you look around, you see some awfully weird things. In fact, you see carbon monoxide twice, 3 times, and 4 times. I keep losing track of the carbon monoxides. And you see 2 cyanides. You all know that cyanide and carbon monoxide are not normally used by our life enzymes. But for this bacteria actually uses them. The reason why they're not used, by the way, is that they stick to our iron too tightly and they mess up our enzymes. So this iron clearly can deal with it. In fact, it's probably using the sticking in some way. It's just kind of weird to see these things that use poison for good purposes. They use poison as vitamins is what they're actually doing here. But on the other hand, to these guys, O2 is poison. And that's the idea of when that first happened, there was this double-edged sword going on. Who has taken 3540? I know Jeffrey has. And Jocelyn, you've taken 3540, and Rianne. So if you ever get the chance to take 3540, you'll talk, part of that is about this. Oh, and by the way, there's another inorganic chemistry course beyond 3540 called Transition Metals. It's only offered every 2 years. It will be offered next year. So if you all want to come around next spring, you could take that, but honestly, I'll give you just a little bit of the upshot. The upshot of that is that you look at the different metals and you see how they react. They each like to bind their ligands. They stick to them with particular geometries based on their orbitals and their place on the periodic table. And so you can do a line like this. Now, this looks like a lot of information, but to RJP Williams, he was an inorganic chemist working with these from the beginning. He just sort of knew, well, this is what iron does. Iron sticks to cyanide, like we had. Iron sticks to sulfur. Iron forms these chunks. And it's what it does in rocks and in life. And that's a really cool thing, that there's a connection between biology and geology through chemistry in this case. So this is kind of like the cheat sheet for RJP Williams' brain. Like, what kinds of things do these different metals like to stick to, and what kind of things, what way do they like to stick to them? Some of them are octahedral, some of them are tetrahedral. This is all goes back to their, the shapes of their orbitals, and the periodic table determines those. So these personalities are set by inorganic chemistry. So it's like if you need to do a certain kind of chemistry, you can look up and down this lineup of available transition metals, and you can find the best person for the job, the one who fits the personality that you need. So that's why when I had the image of a lineup, I don't say what it is. Anybody know which movie this is? The Usual Suspects. So would you say that it's a movie people should watch? Yeah, it's kind of a good mob movie. Like, some people really love it. I think it's just a good movie. But it's actually, I don't want to give too much away, but there's something distinct about it that is really enjoyable once you get through it, okay? So anyways, There's lots of things about, and it has the image of why are these guys in a lineup? Each of them has a personality, and you're trying to figure out who the major criminal is among them. Okay. So the personalities actually can be applied to different metals, and because there's only so much of those metals around, we usually don't have to go below the first row of the periodic table. And we actually don't need to, because the periodic table is periodic. The stuff under iron will act like iron. The stuff under cobalt will tend to act like cobalt. So really all we need is we need the top row of the transition metals. There's only a few simple rules needed to pick one out. And you have, again, this is RJP Williams, straight, unadulterated. You have the different kinds of things. If you need a copper protein, there are actually certain kinds of characteristics that appear to go along with its personality. Even the protein structure appears to have certain shapes that go along with the shape and binding of the metal ion. So I wonder about doing a protein design project where you take one of these rules and you design it into a protein and then throw all the different metals at it and see what happens. Actually, in a sense, our sidericalin project is almost along these lines. When we try iron and we try cobalt, we're trying things with different personalities. And the personalities overlap a little bit, but there usually is a best personality, a best metal for the job. The thing about this is that life will usually find the best metal for the job, and then it will use it for that. Because we had changing metal concentrations over time, there were certain of these, especially 4 and 5, copper and zinc, didn't show up until late in history. So like we had that example of the old enzyme that uses cyanide. Well, here's the examples of new enzymes that use copper and zinc, and those are able to do new things. So it's actually pretty easy to make a enzyme that forms a bond between almost any two atoms, even ones that might be kind of difficult. I mean, if we move on from hydrogen, we have the metals being able to do all sorts of interesting things. Like here's what a nickel surface can do. Nickel will absorb to both hydrogen and to carbon monoxide. And you imagine there's a lot of that around the early Earth. So if you have a nickel surface, the nickel surface can actually absorb to both of those, and then they lean over, they form a bond, and it turns out that that bond is stable. And what you've just done is you have just put hydrogens on a carbon and made this carbon more available. You can do a lot more chemistry with this than you can with that. Okay? So nickel made, and these are also gases. I mean, they're useless to life as gases pretty much. But if you tie them down with a metal, you can actually build things out of them. This is like the little Lego block, the simplest Lego block. And it's formaldehyde is what it is. We don't use it again anymore because it's toxic to us, but that's because it's so reactive. Back in the day, it would have been actually a very useful thing to have around if you were building stuff. You can very easily activate the carbon from the atmosphere if you have a metal. And it looks like this, you know. So really just binding, a surface binding two things, and the two things then bonding each other is a very simple kind of thing. And so when people say, yeah, life chemistry is really complicated and I don't see how it could ever happen. Yeah, if you look at DNA and RNA, I totally agree. That's a really fascinating problem. But if you look at making carbon available and building stuff with carbon, building structures, that actually is not that hard. Okay? In fact, even the biggest thing, the most important thing is oxygen, doing that with oxygen. Now, of course, it's a little bit trickier because you've got to have a source for oxygen, and then you've got to make the oxygen. Remember, the oxygen is a weak bond. That's the whole thing about why is fire hot, right? The oxygen is sort of a weaker end of the double bond, which means it actually will be hard to form because probably the bond you make and the energy you get is not as much as the bond you have to break and the energy you have to put in to make the O2. But this looks like this is a variation of the same theme, except it requires a very specific metal because making the O2 is harder. But it looks like manganese can do this. If you look at how life makes oxygen, It's essentially using a manganese surface. I mean, there's a lot more going on, right? But the heart of what's going on actually looks like that thing I just showed you with nickel, where you have manganese binding water. I like how he writes it, OH2, because what's sticking to the manganese? It's going to be the O. And if you have, the problem is you have to remove the electrons. You have to get the electrons and the H's out of the way to form the bond between the O's. So if you have a removal of 4H and 4E, that takes energy. But once you remove those, they actually can find each other and make an O2. So manganese does not bind sulfide, for one thing. And it actually likes to bind, if we look at the rules here, in fact, let's go back and look at the rules. Right here, Mn is bound by longer bonds to carboxylates and 1 or 2 histidines. If you remember your amino acids, there's no sulfurs in there. Manganese does not like to bind sulfur. But on a more oxygen-rich planet, it would have more things to bind. And really, this is the only thing is that this amazing chemistry is a variant of bind two things and let them bond. So the hard part is that H2O is extremely stable. But at least with it, there's a high concentration. You've got 55 molar solution of this very stable compound. So you can imagine that happening, especially if you can imagine it developing with something next to H2O on the periodic table, which lo and behold, you go down one, H2S would have been very prevalent. And once you have the chemistry for H2S going on, then you already have the shapes of the sites that you need for H2O. This is a way in which even the most amazing life chemistry actually makes inorganic chemistry sense. And that's one of the things that I like to tell people. So you have that same technique going on. Okay, so if I'm sitting here talking about it, I could be waving my hands, and RJP Williams, honestly, he waved his hands a lot. But he said that this looks like it could happen. And so by now, we've had people who've actually done stuff like this in the lab. There's this lab that they took a bacterial photosystem. So this is basically a light catcher. It's the colored molecules, and they catch energy and they funnel it toward a certain part. But it didn't bind manganese, it didn't do photosynthesis. So what she wanted to do is she wanted to put a manganese right there where all the energy was coming down. And she just added asp, glue, and his. By the way, if you look at those rules, what she did was exactly what Williams said you should do. And this was not the hard part. Everything else was the hard part. And then suddenly, it will do the oxygen chemistry only in the presence of light. So if you have light around, it will take that energy and do what we have just on the back end of that. Do you see what it doesn't quite do everything that the last slide shows. It makes superoxide instead of O2. So basically you're making like an extra oxygen-loaded version, like right here. I think you're making H2O2, and that decomposes to make some superoxide. But hey, you're forming a bond between oxygens with very little effort, in my view. So this is, and the fact that it requires light shows that the energy is somehow being used for this process. You don't need much more than a manganese in the right place to be able to do this. In fact, there's people who have taken just the manganese cluster. There's a figure of this in the book. I forget exactly which figure it is. But Gayla Bent drew this picture. And it looks kind of like this. But again, this looks like an iron-sulfur cluster. It's just with manganese, oxygen, and calcium. It's like a little chunk of rock. And the really cool thing is that the calcium actually is important. The relative states of these, this is the best arrangement. It looks like if we mess this up and have just manganese, it doesn't get all the way to oxygen, just like the last slide showed. But it's not that hard to imagine calcium being around and this happening. Also, the other prediction that Williams made is that this should happen outside the cell, or outside the cytoplasm, to be clear. And that's where it happens. It happens in plastids, but actually outside the cytoplasm, because that's where you have more calcium, for one thing, more oxygen as well. So all of these things seem to come about from the stuff the cell ejects, comes together like a rock, and this little rock will be able to actually make superoxide in the last case. In this case, this little rock makes oxygen. And making it honestly, if you had an organic chemistry protocol like this is almost easy enough that I think that I might be able to do it, you know. I've forgotten a lot about organic chemistry, but it's really a one-pot kind of synthesis. You mix stuff together, you get a precursor, and then you recrystallize. And you end up with that. It's a stable configuration of the atoms. Again, just like it's a little rock. Cyclic voltammetry. This is for you, Jocelyn. Okay, so, and you could probably tell me a little more about this. This is another thing I've forgotten. But the main thing is that she did this cyclic voltammetry, and that's how you look for a catalyst, right? You know? And that's how she looked for a catalyst. Or actually, this is a different group. This is Jingquan Zhao. So Dr. Zhao looked for a catalyst this way. But the thing is that you have the metal center, and the metal centers are able to change charge. And one of the reasons why manganese is good for this and other things won't do it is because manganese is going up to +4, and it's going between +4 and +3. Something about those charges and those orbitals make this reaction go. So you wouldn't expect this necessarily to work with iron. You wouldn't expect it to work with another, like nickel or something like that. Something about how manganese is special for doing this. Its personality goes along with it. So because of this, and now I want us to take a step back and look at the big picture. Because of manganese, you have the sunlight energy being turned into chemical bonds. And those chemical bonds can also be broken at another time. In a sense, it's storing the energy, because if you break those bonds at another time, you can get energy out of them. But it's really from energy from very far away, from outside the Earth system, and that's what makes life possible. The sun is hugely important for this whole process. And the sun's output is in joules. So we can think about this like physical chemists, and we can look at the different things. Photosynthesis is basically using this energy to make bonds. Synthesis is synthesizing new chemical bonds. And you can look at plants, and you can say that plants have increased the net joules that have been captured by our planet. Here's a very crude, time scale, 10 to the 9th years, right? This is very different from the kind of kinetics graphs that we're looking at. But without energy capture, you would have a single exponential curve down where the Earth would be, the sunlight would shine on the Earth, but it wouldn't be really captured until you have life deliberately putting it into bonds, deliberately forming structures from that energy. And we benefit from this when we eat them. We eat the plants, we break the bonds, we get the energy. The energy ultimately comes from the sun. I like this graph because it shows sort of a kinetic decay, but it shows that once photosynthesis developed, this is a much more complex kinetic mechanism. We started to have a positive slope to that. Started to be able to capture energy. Another delta G graph to look at it is that when you have these things, and remember, heats of formation, free energies of formation, they don't care about the state that they're in, and so you can look at the whole thing about life as being, life starts at these stable molecules, like CO into H2O, this is ancient life, and then energy comes in, and it gets stored in chemical bonds. Originally, that energy could be stored in the Earth's own bonds, and you could break those bonds and make other bonds. But that's going to run out of energy pretty fast. And there's not as much energy in there as there is in the sunlight. Sunlight can push you up here, and you can sit up here. For as long as you can protect your bonds from degrading, you can sit up here, and you can basically save until you need energy, and then you break the bonds, and you use the energy to move or to have vision, to take in information, to think even. So that thing about chemical bonds storing energy, you are in a less stable state when you form this, and most of life's structures are actually less stable, but that's why you get energy by breathing out CO2. You're making it more stable, and the more stable molecule is the dead molecule. The molecule that's useful for life is actually the less stable molecule, because then life can choose when it uses its energy. Okay, so it's imagine the sunlight actually gives you delta G, and take a step back, second law of thermodynamics is still being followed. Because the sunlight is high energy, you can even take the wavelength of the light, and the light going out is dispersed energy. It's called heat right here, but it's probably better to say what form of energy is heat energy leaving in? Infrared. So you're taking like visible light, and you're stretching it out to infrared, and that's what life's doing when you sit back. That's increasing entropy, because you're stretching something out. And that's why the second law of thermodynamics can be obeyed despite the fact that you're sitting in an unstable state. You're using this energy, this entropy increase, to keep yourself at the stable state. The unstable state, I should say. Okay. So here's something from the exploratorium. This is kind of cool. They have this growing in the sunlight in the San Francisco Bay Area Science Museum. And they actually have a little flap that they've kept on. You can undo the flap, and you can see there are different bacteria behind there. There's no green bacteria behind there. The green is to catch the visible sunlight. And actually, they have some old purple sulfur bacteria, so they have a big sample of these weird purple sulfur bacteria. In these, and I believe this is, at least this is deep in the mud, so there's not much oxygen getting here. Okay, they actually are they're breaking apart chemicals like sulfur and methane is what they show. You still have bonds changing, it's just the bonds are less energetic. But that's why, if you look at the energies and you add them up and you see how much energy we're using, it just doesn't add up without oxygen. That's one of the arguments of the book. If you move on to life with oxygen, cyanobacteria, they even have a color in the name. And so that's why we had all those readings about colors, and you can literally see that in the exhibit. You can see the life is getting more colorful. And so this is probably the central graph of the whole book. That as you have billions of years go by, oh, and by the way, remember that previous graph that we had energy storage being increased. This is part of that. As oxygen concentrations increase, this is one of the ways that the energy was stored on the planet. It was stored in the form of O2 bonds that could later be combined, broken apart, combusted. So you have this whole thing, this whole graph, and you've read about this. You have the geological consequence of the banded iron formations, which is iron and oxygen. So I also want to remind you of this graph. This is probably the other central graph to the whole thing. Notice how there's the rightward shift on the periodic table away from using manganese, iron, copper, nickel toward using more copper and zinc, cobalt, nickel. This is a version of this, and notice how I had it as an arrow because the oxygen is going up. Therefore, the oxidation potential, the redox potential, is changing in one direction. So time changing one direction, why not make it an arrow? I'm going to show you one of the most inexplicable to me things that RJP Williams did. This is his version of the graph. I mean, it looks kind of cool, but it does not give the impression of a graph. Like you have a great x-axis that's either time, or oxidation, or things like that. Instead, what he did is he just took the E values. So now that we've talked about the E values, you can sort of see what he did. You have a question about it? Yeah, me neither. Maybe it's an inorganic chemistry thing that they did. It's actually two-sided, and he puts all the metals over here and the non-metals over here, and he talks about each of the transitions. Each of these is an E value. So, and he shows you that 4 times 10 to the 9th years ago, yeah, we were a reduced planet, and the E value of the planet was down about here, about hydrogen. Now we have a lot more oxygen, and the E value, that is the active component, the active redox component of our ecosystem, and we have moved to this E value. And as we've moved, we've flipped these from the reduced side to the non-reduced side as the oxygen increases. He does have arrows, but my goodness, I knew I had to do something with this. So this is, if anybody ever sees a graph like this from our inorganic chemistry textbook or something like that, maybe it'll help explain it, but this is a mystery to me. But the main thing is you can see that this does predict that you're going to have these changes over time. Nitrate will become ammonium. Sulfate will become sulfide. And carbon dioxide will let's say right here, that actually carbon dioxide can be turned into carbon structures, but you actually are moving from, this one is a little bit more confusing because we actually don't want to compare this to formaldehyde. We don't really care about that. We want to compare it to methane or CO. And actually, you have that transition up here, where it's going to be moving from the CH4 to the CO2. You're just going to end up with more of the oxygen-bound form as oxygen increases. But you have to dig through this graph to get that thing. And the other side of things, instead of having O's appearing and H's going away, you end up with things going from the reduced form to the oxidized form. You end up with electrons being pulled away from things. Iron 2 became more rare, and the fundamental thing, every chemist knows this, iron 3 hydroxide is very insoluble. So if you mess things up with your experiment and you have iron 2 in there, it will actually become unusable. And in a sense, the experiment of the planet, the iron in it became unusable when oxygen showed up. This is what I really want to show you with this whole talk. This is the data from one of the 3 groups of scientists that looked at genes, and they said, when were these different elements used? And so if you look up and down here, this actually is similar to the RJP Williams predictions, which came out way before this paper. And they measured whether there was more or less of the different molecules being used by the genomes of all life. So the one thing that you can see, you can sort of calibrate yourself to oxygen. Oxygen went from blue to white, which means it went up in gene fraction of things that used oxygen. So basically, blue is low, red is a lot. And so something that's starting red and going down, so we can go up and down, we can see these data. Which of these data go with Williams' predictions? Well, here's the SO4 goes from blue to white. That's going up. The H2S goes from red to white. It's going down. Copper goes up over time. Molybdenum also goes up over time. Copper and molybdenum. Molybdenum is the third thing that you mentioned after copper and zinc go up. Manganese goes down. Again, that's on the left side of the periodic table. Cobalt. It's a little bit of a weird blip back here, but that looks like a data issue. It definitely looks like we have a lot more orange, and then it's going down. I'll definitely count that as a plus. Zinc is weird, but actually zinc has always been weird, because zinc is actually used by the ribosome a lot, but it's used in a different way. It's not used really in a catalytic way. It's kind of a structural way, and zinc is just kind of weird. And so zinc throws a little bit of a wrench into the simple statement. And their data for nickel show that nickel's actually going up, which is the opposite of what Williams predicted. So there is one thing that he didn't, well, actually, but they don't trust their own data when it comes to that. So I don't want to say that he got this wrong. I want to say that the story is more complicated. And we also have another good story about nickel, the nickel famine, which really sort of is another data source that would be right along with these predictions. So this data is weird, but it's weird with all the other data. And iron's weird, but it's also, remember, iron is really unique in its properties, and so it's really fought over by life. In fact, that's the whole point of siderophores, is that they fight over iron. So it's possible that iron is decoupled from the environmental concentrations of it. Once life got sort of hooked on iron, it kept scraping up the iron wherever it could find it. So things became tough for life, but life evolved ways to keep using iron. And honestly, the iron is just, it's just basically white, so it looks like a null value. So if you probably told the RJP Williams, and this doesn't even talk about the nitrogen and carbon follow the same general patterns. The general pattern is the H's are red, and the more oxygens you have, the more blue you are. And that's sort of what you see. Okay. Yeah, same. They represent how much, I think they represent how often this was seen in the genes. And so that might be, for example, iron's used a ton, and you see it has a high number, and zinc is used a ton. Copper is used less, and so it has a lower number. So it tells you a little bit about the prevalence. It's possible because nickel's numbers are so low, that's why it's not giving good data. Also, yeah, like N2 is a little bit weird, and it also has low numbers, although, yeah, N2 is kind of weird. Actually, I would say that N2 fits a prediction as well. Basically, nobody's, at least as far as I know of, nobody's tried to redo this because it just came out to be along the lines of predictions. I wonder what would happen if we redid it today, if some of these blips would go away, or if we'd get more blips. But the bottom line is, if you're like right 9 times out of 10, you're pretty much right. Okay. This is another graph that they had in their supplementary figures. And here we have our nice time axis. This is showing when oxygen-using genes were invented, when they were first seen. What's really interesting, so you see the peak right there, right after 3.0. The magic point when we first see photosynthesis happening is right around 2.9, 3.0. So photosynthesis was invented right here, and within 0.1 billion years, you have a bunch of oxygen genes being invented. This is what we mean when we say the data supports Williams. Okay. So after you had oxygen, you could have enzymes doing all sorts of cool new things. We have evidence for these enzymes being invented after oxygen, so 3.0 billion years ago or later. And again, this was written by RJP Williams 10 years before they came out with the data. But this is what the data shows. You could do these new things. Now notice that molybdenum actually doesn't show up in the same way on those things. It's a little bit more complicated because it has a lot of oxidation state. But it changed its oxidation state to become higher, and its higher oxidation state is the more useful form that can do more things. So, and actually, like I said, molybdenum is the third advanced metal. You know, when oxygen came about, it released copper, zinc, and molybdenum. And I have to show you this one because it shows you literally a redox potential range for different things of life. And you'll notice that this is inside the cytoplasm. Now the cytoplasm, even your cytoplasms, tend to be reducing. But the other compartments of the cell are not as reducing. Obviously, blood is very oxidized, right? Because it's oxygenated. But if you look at the redox ranges that you see inside anaerobes, you see a lot of negative volt values. So below the H2 standard potential. And as you go along, if you look at things that use iron 3, same thing. Sulfate users, you see a few things that can handle a little bit higher oxidation. Nitrate users, they have a higher oxidation, and then us go all the way up to 0.8 volts, which is exactly the E value of O2. So it's like we can handle, this is evidence that it really is the O2 that's controlling things, and that these things developed, these chemistries developed in this order, okay? It's an interesting kind of graph to imagine, making a graph with volts on the axis. But honestly, volts on the x-axis and years on the x-axis are going to be very similar. So using an electrode to measure potential, one way to measure the time of origin. The confounding factor here is that most of our cytoplasms are still look like an anaerobic cytoplasm. It's kind of weird to think about. Just like you have mitochondria giving you energy, you have most of your cytoplasm is an ancient sort of recipe. So the other thing is that you can do sequence of availability. There's actually a connection between E and KSP as well. But the main thing is as the Earth goes from H2 to O2, H2 has a 0.0 volts, and O2 has a plus 0.8 volts. You can imagine the whole planet changing its E values, which means at equilibrium, there will basically be more oxidized, more of the oxidized side of each thing will be, all these equilibria shifted. So I just want to show you, this is his version of the same kind of tables that you know and love from chapter 5. But here he is doing the exact calculations that you're doing in PChem. And he's using this to show, yeah, if you aren't at equilibrium, if you aren't at the standard state, it's going to shift your equilibrium this way. That's why that's why I make you do that homework, okay? Because he uses it to make his argument. So the idea is that, yeah, at first you had this chemistry available, and the chemistry outside the box was not available. And oxygen expanded the box of what you could do. There's one more thing that that's really interesting. Never just think that a molecule can do one thing. Usually you see it doing multiple things. So I say quickly that oxygen was useful to making cholesterol, and there's a version, I think there's a version of this graph in there. I might have cut it. But there's a version of this reaction, and notice this is a reaction synthesis. It starts with a carbon chain and ends with cholesterol, and it goes through oxygen. It requires oxygen as a reactant. You couldn't do this 3 billion years ago. You had to wait until like 2.5. But once you have this, the cells can do a lot more things with the cholesterol. The thing that's usually cited is that they can be much more flexible, they can be stronger. The way they mix into the membrane makes it stronger and more useful. But there's also some people that argue that it's not just that. Cholesterol has a chemical activity as well. So this goes with the idea that asteroids have hidden chemical reactivities or chemical activities. They can catalyze helpful reactions. Cholesterol might be able to catalyze a helpful reaction in the sense of, well, not really in the sense of catalysis, but in the sense of it's good for protecting the cell. If you have more cholesterol, the cell will survive. The thing is, without cholesterol, the membrane sort of looks like this. It's more wiggly, right? If you have cholesterol, then you have a membrane, the membrane holds together better, and it stacks together better. And that means that if you have any kind of degrading chemical, it won't be able to get as deep in. It's going to bounce off. It's going to be a better wall for the cell to keep the degrading chemicals out. And what is the main degrading chemical that they're worried about? Oxygen. So oxygen allowed the creation of cholesterol, which helped protect the cell from oxygen. When you have two things that are helping the cell at the same time, you have an additional survival advantage. And so that makes something that looks like, oh, it looks like evolution couldn't do this, right? Well, if cholesterol actually can do double duty and it gives you two advantages rather than just one, then maybe it could happen, you know? So that ends our data talk. So I'd like to have a little stretch break. As we do a stretch